Assalamualaikum warahmatullahi wabarakatuh. My name is Sri Ayu Limbong Minanga as a member of Group 13. On this occasion, I will continue with material related to plant biotechnology. Let's get started. So what is the meaning of biotechnology? In general, biotechnology is the application of biology organism, system and engineering process in industrial goods and services that are used for human benefit. Biotechnology comes from the word bio, which means life, and technology. So it can be concluded that biotechnology is a technology that utilizes microorganisms to products and services for the benefit of humans. Next, biotechnology plays an important role in technology achievement in human history. Biotechnology is one of the fast-growing applied science following the demands of globalization. In general, biotechnology is divided into the two types, namely classical or conventional biotechnology and modern biotechnology. The characteristics of biotechnology are, first, the existence of biotechnological agents in form of plant or animal microorganisms. The second, there is utilization in technology and industry. And then, the resulting product is in the form of extraction and refining. There are so many fields related to biotechnology, one of which is plant biotechnology. So, what is the meaning of plant biotechnology? Plant biotechnology is a part of biotechnology that focuses on the field of plants. Plant biotechnology has played a role in producing food plants with higher nutritional contents. Plant the products useful compounds so that they can be used as herbal medical plants that are resistant to pests and so on. There are so many examples of the implementation of plant biotechnology, especially those related to agriculture, include tissue culture, hydroponics, and genetic engineering. Tissue culture itself is related to genetic engineering because genetic engineering is mostly carried out in vitro in the laboratory. Okay, in this video, I will only discuss the example of the implementation of plant biotechnology, namely the first, hydroponics. So, what is the meaning of hydroponics? Hydroponics comes from the Greek word hydro, which means water, and ponos, which means work. So, hydroponics means working on the water or working with water. The method used in hydroponics include the water culture method, using water media, and then the sun culture method, and the porous method, or using grave media, broken branch, and other. There are so many examples of plants that can be hydroponic, such as aglaonema, dicrana, tomatoes, peppers, mustard, greens, kale, lettuce, melon, and so on. Okay, this is the advantage of farming with hydroponics. Consists of, first, plant can be cultivated in a place, and the second, there is no risk of crop damage from lack of water and erosion. 3. No need for too large a land. And then 4. Plant growth in faster. And then 5. Free from pests. And 6. The result are high quality and high quality. So this is a picture about example of hydroponics. So this slide is the second part of an example of the implementation of plant biotechnology, which I will discuss, namely general manipulation. So definition of genetic engineering. Genetic engineering is an activity of gene manipulation to attain new products by making recombination DNA through gene instruction. Recombination DNA is DNA whose sequences have been recombined in order to have the desired properties or function. Next, there are two components involved in genetic engineering, among others. First, plasmids. Plasmids are dual ring-shaped DNA molecules that are specially found in the bacteria. In genetic engineering, plasmids function as vectors used to transfer and reproduce foreign genes. And the second, enzyme. Enzymes play a very important role in genetic engineering. In the process of the genetic engineering, there are two types of enzymes that are directly involved, namely: 1. Endonuclease reaction enzyme. This enzyme functions to cut the DNA chain, so it's known as biological scissors. This enzyme has the ability to record signs and cut certain nucleic sequences in DNA. And then two, ligase enzyme. Ligase enzyme serve to glue the DNA fragments. Okay, this is 4 kinds of genetic engineering technique. First, plasmid technique. Plasmids are called genes found in bacterial cells unrelated to chromosomes. Through these techniques, experts in biotechnology can develop transgenic plants that are resistant to pests and diseases, to dry and less fertile soils. And then 2, hybridome technique. Hybridome technique is the joining of two cells from the same organism or cells from different organisms, resulting in the single cell which has a combination of the two cells. And then 3, gene therapy. Gene therapy is the repair of genetic disorders by repairing genes. This gene therapy is usually used to fatal diseases such as cancer. And then 4, cloning technique. Clone comes from the ancient Greek words "alon," which means a twig or a grape. The term cloning is used to describe a group of living things that were born without a sexual process, along with the development of more advanced biotechnology. Various transgenic plants have now been produced, for example, such as anti-pest plants. So this is picture of, for example, genetic engineering. This slide we can see this is watermelon, where generally watermelon have a red color, but in genetic engineering the color of the watermelon able change with blue color. So this picture refers to watermelon have a blue color. Okay, I think that's all. And assalamualaikum warahmatullahi